Next, we'll move on to autophagy. So, auto means self, phagy means eating. It means self-eating. So, during conditions of some stress, the cell is going to eat up its own organelles to survive the stress. Okay, so it is also called as cell cannibalism, and it is a survival mechanism. So, it can happen both in physiological and pathological states. So, example, in aging and during exercise, while in pathological states, usually in cancers, malnutrition, and during starvation states, autophagy will happen. Okay, and the organelle involved in autophagy will be a lysosome. So, lysosome is the one which is a degrading machinery, right? So, the organelles will be taken up to the lysosome and the organelles will be degraded and their energy from them will be taken. So, this is also a programmed cell death only because it is regulated by certain genes called as ATG genes. ATG stands for autophagy. So, ATG genes regulate them. So, it is a programmed cell death. The most important autophagy gene will be a ATG1 gene. So, 1 for first. So, ATG1 gene is the most important one. And here, this can come as an image-based question also. He is scientist Oshinori Osumi. He gave the pathogenesis of this autophagy and he was awarded the Nobel Prize in 2016 for the same. So, let's see about the types of autophagy. So, we have 4 main types of autophagy, starting with macroautophagy. Macroautophagy is the one in which there is going to be a membrane formation around the target organelle. Suppose the cell wants to eat up the endoplasmic reticulum and the mitochondria. So, it is going to throw up a membrane around this endoplasmic reticulum and mitochondria. So, it is being marked by a boundary, and those organelles which are being marked by this membrane will be degraded in the lysosome. Okay, so there is formation of this autophagolysosome which will encircle the target organelles in macroautophagy. It involves 3 steps. First is initiation phase. Next will be the nucleation phase, wherein the membrane is going to start forming. Then elongation and maturation, so that this membrane formation is complete around the target organelle. Okay, so there was a stress like malnutrition, which is going to initiate the formation of the pre-autophagosomal structure. So this is the initiation phase. So pre-autophagosomal structure, PAS structure is going to be formed. So this is actually supported by a complex called as ULK1 complex. So ULK1 complex will associate with autophagy genes ATG, and then it will form this pre-autophagosomal structure. Then what happens? This pre-autophagosomal structure will then form a phagophore. So phagophore is nothing but an isolation membrane. So the membrane is going to start forming. So this membrane can be acquired either from a endoplasmic reticulum or a plasma membrane. So this starting of the membrane formation is called as nucleation phase, and this is going to be caused by a protein called as Beclin-1. So this Beclin-1 protein is again going to associate with autophagy genes, and it is going to form this phagophore. So the next step will be from this phagophore, this membrane is going to form a complete round around the target organelle. So the center organelle is the target organelle, and this isolation membrane, which was like this, had formed a complete vesicle around this target organelle. So it is called as the autophagosome, which happens in the elongation and maturation step. So for this to have autophagosome to form, what happens is this Beclin-1 complex, which was formed in the phagophore, it is going to cause covalent linking of 2 proteins called as phosphatidyl ethanolamine and a microtubule associated light chain 3. So this in short form we call it as PELC3. So this PELC3 association is going to be caused by this Beclin complex. So this PELC3 formation will result in the formation of the autophagosome. Then this autophagosome is now going to the lysosome and fuses with the lysosome. So this autophagolysosome fusion will take place, and inside the lysosome the target organelle will be degraded. Okay. So we have this is a very new concept over here. So there are potential MCQs in this. There is a protein called as mTOR protein. So this mTOR protein is going to inhibit this ULK1 complex. So what will happen? This mTOR is actually inhibiting autophagy. So mTOR will inhibit autophagy. While there is a rapamycin, a drug called as rapamycin, which is going to inhibit this mTOR. So rapamycin is actually promoting autophagy. Okay. Then there is BCL2 protein, which was a anti-apoptotic protein, right? So BCL2 is going to inhibit this Becklin. So B for B, so BCL2 will inhibit this Becklin complex, and that will actually inhibit autophagy. So mTOR and BCL2 are going to inhibit autophagy. So the PLC3, which is the final step which resulted in the formation of this autophagosome, is called as the marker of autophagy. So marker of autophagy is PLC3. Okay. Next, we'll move on to mitophagy. So as the name suggests, here the target organelle is going to be mitochondria. So it is a form of macroautophagy only, but the target organelle here will be a mitochondria. And what are the genes involved here? So we have Parkin gene and PINK1 gene. So Parkin and PINK1 genes are involved in mitophagy, and when this mitophagy is being impaired, it is going to lead to a neurodegenerative condition called as Parkinson disease. Okay, Parkinson's disease, it involves, the pathogenesis involves this Parkin1 and Parkin and PINK1 genes. These are actually involved in mitophagy. So Parkinson's has impaired mitophagy. Then we'll see about microautophagy. Third type of autophagy is microautophagy. It is not involving any of this formation of target organelle membrane. So here it is just endocytosis by the lysosomes. So this was the lysosome. The lysosomal membrane will just form a cavity like this, and it will take whatever cargo is coming and then close it and digest it. Okay. So this endocytosis by lysosomes is the mechanism by which microautophagy happens. Then chaperone mediated autophagy. So chaperones are nothing but foldases. So they help in the proper folding of the proteins. So HSP17, which is heat shock protein 17, is one of the chaperones. So when there is a misfolded protein, this HSP17 70 is going to associate with it, and it will try to cause the proper folding of this protein. But when this proper folding is not possible, this misfolded protein has to undergo degradation. So what will happen? This HSP70 will take up this misfolded protein and take it to the lysosomal membrane. So in the lysosomal membrane, we have a protein called as LAMP2A. So LAMP2A is nothing but lysosome associated membrane protein 2A. So via this LAMP2A, this misfolded protein will enter into the lysosome, and there it is going to be degraded. So this kind of autophagy is called as chaperone mediated autophagy, which helps in the degradation of the misfolded proteins. So there are certain diseases related with autophagy. There is a susceptibility for, if the patient is having autophagy 16 ligand single nucleotide polymorphism. So in that, it will lead to Crohn's disease. So this kind of susceptibility will lead to Crohn's disease more than ulcerative colitis. So usually it is Crohn's disease. Okay. Then if there is ATG5 deletion, so the patient is going to have an increased risk of tuberculosis.